Cancer results from uncontrolled cell division, leading to the formation of malignant tumors, which are masses of abnormal cells that can invade or metastasize other tissues in the body. By contrast, a benign tumor always remains at the original site. The term transformation refers to a process by which cells acquire the properties of cancer. There are three classes of defects that can contribute to cancer transformation: oncogenes, mutated tumor suppressor genes, and mutated stability genes. Proto-oncogenes are normal genes involved in cell growth and division. The conversion of a proto-oncogene to an oncogene can cause overactive cell division and lead to cancer. The mutations that produce oncogenes are genetically dominant, meaning that tumors form if one of the homologous chromosomes contain genetic defect. Examples of oncogenes include the gene that encodes for G protein RAS that play a central role in the signal transduction pathways of many growth factors. As I covered in my previous video, RAS is part of the MAPK pathway that is activated by GTP and ultimately activate gene expression for cell division proteins. The RAS oncogene encodes a protein with normal GTP binding activity, but no intrinsic GTPase activity to terminate the active state. The mutant RAS protein is therefore always activated regardless of the signals arriving through normal receptors, resulting in uncontrolled cell division. Mutations in RAS are associated with 30 to 50% of lung and colon carcinomas, and more than 90% of pancreatic carcinomas. The second class of defect that contribute to cancer involves mutation of tumor suppressor genes, which encode proteins that normally restrain cell division. Unlike oncogenes, defector tumor suppressor genes are genetically recessive, meaning that tumors form only if both chromosomes of a pair contain a defective gene. Examples of tumor suppressor genes include p53, which function in regulating cell cycle and apoptosis, or program cell death. Mutations in p53 are responsible for more than 90% of human cutaneous squamous carcinomas, also known as skin cancers, as well as 50% of all other human cancers. The third type of defect involves mutation of stability genes, which encode proteins that normally function in the repair of major genetic defects. Mutation of stability genes would lead to accumulation of unrepaired damages in other genes, including proto-oncogenes and tumor suppressor genes, and thus lead to cancer. Examples of stability genes include the XP gene, which functions in nucleotide excision repair to fix UV-induced pyrimidine dimers. Mutated XP genes lead to skin disorder known as scleroderma pigmentosum, in which patients are highly susceptible to sunburn and skin cancer. Another example of stability gene is BRCA1 and BRCA2 genes, which normally repair double-strand breaks. Mutated BRCA1 and BRCA2 genes will lead to increased chance of breast cancer. Mutation in the HNPCC gene, which normally codes for proteins involved in the mismatch repair mechanism, will lead to higher chance of developing colorectal cancer. Typically, mutations in oncogenes and tumor suppressor genes do not have an all or none effect. The progression from a normal cell to a malignant tumor often requires an accumulation of mutation. For example, the development of colorectal cancer has several recognizable stages. First, the epithelial cell in the colon undergoes mutation of both copies of the tumor suppressor gene APC, which stands for adenomatous polyposis coli, resulting in uncontrolled cell division of epithelial cell, leading to a small benign tumor known as a polyp. Next, the APC gene causes chromosomal instability that can lead to another mutation in the oncogene RAS, converting the polyp into a larger tumor known as adenoma. Further mutation in the tumor suppressor gene DCC leads to late adenoma. Finally, only when both copies of the tumor suppressor gene p53 become defective does the adenoma develop into a carcinoma, a malignant life-threatening tumor. In addition to mutations and genetic alterations, a number of tumor viruses account for 15% of cancer cases worldwide. Major tumor viruses include Epstein-Barr virus, abbreviated as EBV, which has been linked to Burkitt's lymphoma, a cancer of B lymphocyte in the lymphatic system. Human T lymphotropic virus, abbreviated as HTLV1, is associated with cancer of the T lymphocyte. Human papilloma virus, abbreviated as HPV, is associated with cervical cancer. Cancers can be classified into 6 categories based on tissue types. Carcinoma originates in the epithelial tissues, which covers our skin and lines our internal organs. Carcinomas account for 80 to 90% of all cancer cases and usually affect organs capable of secretion, including breast, lungs, colon, bladder, and prostate. They are divided into 2 major subtypes: adenocarcinoma, which develops in an organ or gland, and squamous cell carcinoma, which develops in the squamous epithelium. Sarcoma originates in the connective and supportive tissues, including bones, tendons, cartilage, muscle, and fat. Examples of sarcoma include osteosarcoma, which affects the bone, chondrosarcoma, which affects the cartilage, leiomyosarcoma, which affects the smooth muscle, rhabdomyosarcoma, which affects the skeletal muscle, mesothelioma, which affects the membranous lining of body cavities, fibrosarcoma, which affects the fibrous tissue, angiosarcoma, which affects the blood vessels, liposarcoma, which affects the adipose tissue, glioma, which affects glial cells of the brain, myxosarcoma, which affects primitive embryonic connective tissue, and mesenchymal mesodermal tumor, which affects mixed connective tissue types. Lymphoma originates in the glands or nodes of the lymphatic system, which is a network of vessels, nodes, and organs that purify bodily fluids and produce immune cells. They are known as solid cancers that can develop at specific sites such as stomach, brain, and intestines. Leukemia originates in the bone marrow, which is the site of blood cell production. They are also known as blood cancers and liquid cancers. Types of leukemia include myelocytic leukemia, which affects blood cells developed from myeloid lineage, such as neutrophils, erythrocytes, and macrophages. Lymphocytic leukemia affects blood cells developed from lymphoid lineage, including lymphocytes and natural killer cells. Erythemia mainly affects red blood cells. Myeloma originates in the plasma cells of bone marrow, which produce various antibodies in response to infections. The last type of cancer is mixed type from different categories, such as adenosquamous carcinoma and carcinosarcoma.